before we jump into the rest of stoichiometry, let's take an aside and talk about an analytical technique that's used to help determine the molecular weights of compounds, and that's called mass spectrometry. All right, so the way this works is that we take a sample, okay, and the sample is usually in the gas phase, or it could potentially be in the liquid phase, and what we do is boil it so that way it becomes a gas, and what we're doing is pushing this gas into a tube. And once it's inside the tube, what we then do is push it in front of an electron beam shower. So what's going on is that as this molecule is going past, you have all these little electrons, which are very high powered. It's almost like you're going in front of a shotgun of electrons. And so these high powered electrons, what they're doing is starting to fragment off other parts of the molecule. Now, it could be whole fragments of the molecules. It could be electrons, could be protons. It all depends. Okay. So as this molecule goes through this electron beam where it's being showered with these high-powered electrons, what it then does is goes through a series of accelerating plates, so that way these molecular ions are speeding up through these tubes, and then it goes through a magnet, passes through a magnet. And what happens is that once it goes through that electromagnet, what happens is that the ions start to separate themselves based on their sizes. So eventually, when these molecular ions start hitting our detecting screen, they're going to be separated out based on their size, their mass to charge ratio. And so once the molecules do that, here's what we get back. We get a graph. Okay, so this one's not terribly exciting, but this tells us a whole lot of information. So the very first experiment that was done in around 1920 was the separation of neon. So what was done in this experiment, though, is that a sample of neon was injected into the system, and then we saw the result was that there were 3 different peaks. So the majority of neon came out as that had an atomic mass of 20, okay, and that was about 90.92%. We had about 8.82% abundance at neon 22, but then you had this thing at neon 21, 0.26%. Okay, this was the first time that anyone had actually thought of something called an isotope, where you have the same atomic number but differing atomic mass. And at this point in time in the story of chemistry, neutrons are about 12 years away from being discovered. All right, so this is actually a really cool technique. And as you go forward in chemistry, you're actually going to talk about mass spectrometry a lot. So right now in terms of chemistry, there is actually 4 ways to use a mass spec. Okay, so there's 4 different ways of using a mass spec. Okay, so let's talk about them. The first way is to determine the molecular weight of a sample, just like what we're doing here. Second way, also what we're doing here, talking about the presence of isotopes. Okay. Now, the third way, this is if you go forward in chemistry and take organic chemistry, you'll actually spend a lot of time talking about mass spectrometry and how this is used. But what happens is that if we know the fragmentation patterns, we can actually get a 3-dimensional structure of a molecule. So if we study the fragmentation patterns, we can get a structure for that molecule. Okay. And then finally, if you make it past organic chemistry and go on to biochemistry, mass spectrometry is used to sequence nucleic acids and proteins. Okay, so uh, mass spectrometry really came into being definitely in biochemistry with the advent of the Human Genome Project, which was started in 1986. And what it was meant to do was to sequence the entire DNA sequence of humans. That project should have taken about 30 years to complete, but the project was actually done in the year 2000, where a rough draft was actually submitted. And the reason why it was done so quickly, 14 years, out of 30, was that because of technology like mass spectrometry, which allows proteins and allows nucleic acids to be sequenced pretty accurately, pretty quickly.